My question is for Dr. Bayard. Great presentation. I'm wondering if, since you isolated the organisms to pure culture, if a different technique like MALDI would be equivalent to what you've done, and I'd be interested in your insights on why you would not use MALDI for your application.
Thank you for your question. I think MALDI is a great technique. It goes also below species level, but I'm not sure if for all of the species you will have the same discriminatory level. Also, the technology is quite expensive. You need to have a good database, and these are all things which would hamper me to use the MALDI. Also, with whole genome sequencing, as it was also mentioned by other presenters, it is a protocol that can be used for different microorganisms. So it was also said for some Salmonella serotypes, you can use one technology, but then for another serotype, you cannot use the same because of the discriminatory power. Also, I showed an example of Salmonella, but for Listeria monocytogenes, you could have used the same approach. So that makes it very interesting as well.
Yeah, I'm Mieke Uitendaal. I come from Ghent University in Belgium, Department of Food Safety, Food Quality. I'm not into whole genome sequencing that much, but I would be a user or interested. But I'm into lab accreditation a bit, and also in ISO. So my question is more, I think the first speaker and the last speaker quickly referred that if you have the sequences, you have to compare them with a reference strain or a reference genome. So I'm wondering a bit, who and why do you decide, or who decides on that reference genome? Is this harmonized? If you use another, if you pick another reference strain, will you get the same outcome? Yeah, this is what this as would interest me. I mean, if several labs, I mean, now it's in an FDA and in your public health lab, but if this goes out to commercial labs and I'm a user and, okay, I send the strain, how am I sure that I get the same, that they're doing it in the proper way?
So the question is about reference genomes and how we select to use which ones, which I think is a fantastic question, and everybody up here has had to wrestle with that issue. And in many cases, it depends on the sample set that you're looking at as far as which reference you want to use. But I do know that there has been a fair amount of discussion about identifying what reference genomes may be used for any particular bug. For within the TB world, really the decision as to which reference to use has certainly been ongoing, and I've been part of those conversations as well. In other specialized organism worlds, as it were, certainly there's a lot of discussion about which one we want to use as a reference, so everybody's comparing to the same one. But if your question is about identifying a point source outbreak, you may want to use a reference that is closer to the strains that you're looking at in your outbreak, so that you can get better resolution. You'll identify more SNPs. So it's not always the same. It really depends on the question you're trying to ask. Sometimes we've worked on outbreaks where there is no reference because no one's ever sequenced this species before, and we're going to do more and more of that as we continue to use whole genome, especially in the fungal world. There's not a lot of in fact, there's pretty much no references to use except in very extreme situations. So I think it really depends on the question that's being asked. But from our perspective, we're happy to work with other people's references and compare our data, but we also try to find a better reference genome for a particular data set depending on the question.
Does this mean that you need to be a reference lab to have access to all this kind of reference?
No, so the reference in this context is not like a reference lab. It's saying this is the baseline, and you compare everything else in your data set to that reference. A reference does not, in the case of the Nestle presentation, the reference was a PacBio sequence, but you don't necessarily have to have a full, complete genome without any gaps to serve as a reference. You could use a draft quality sequence as a reference. And that what we've seen at FDA, and this is the work of Errol Strain in the bioinformatics group, is that the further out you go, if your reference is further out phylogenetically from your set of interest, the more SNPs you're going to detect, which a lot of those SNPs might not be of very much concern. So really, he found that if you're within the outbreak clade, or even the same PFG pattern, is the level of distance you want in your reference. If you go out further to serotype or to species, you're going to get a lot of SNPs that are not going to be very interesting.
I guess on a related note, what is also important with whole genome sequencing is you need to have a quality assurance system in place. And we are, of course, very well aware of that. But the first thing that we need to agree on, and there is no international consensus about that, is what is a good sequence that you can go on analyzing. And we need to define the parameters that we internationally are going to use as criteria for sequences that we can continue working on. And at the next level, each laboratory that are going to do whole genome sequencing, they also need to be accredited or certified. And those systems have not either been set up internationally yet. We have a system in place in PulseNet, and we have one with Genome Tracker. And we're trying to harmonize those two, so we only have one system here in public health in the United States, but we need to have systems in place also internationally.
I know for at ECDC they're setting up a proficiency trial for whole genome sequencing, and Public Health England has signed up for that, and they're going to send out isolates for the whole sort of process from extracting DNA to doing the bioinformatic analysis. They're also going to send out extracted DNA, and they're going to send out genome sequences for bioinformatics analysis. So they're trying to sort of look at how different laboratories in different countries are analysing the data and seeing if we do get the same results. And that's to help in sort of assuring quality, and it'll certainly help in accreditation.
I just want to talk a little bit about what we're doing at CIFSAN. So last year we sent out a proficiency set that consisted of 4 Salmonella and 4 Listeria strains that had been completely sequenced with the PacBio, so we knew what the definitive sequence was. And we, as with the ECDC, we sent out both DNA and cultures. This year we've expanded that to include Campylobacter strains, Shigella, E. coli, and an additional Salmonella strain. So those are going to be going out in August. Again, DNA and, I'm sorry, this time it's just cultures. And another thing I wanted to mention is that within the U.S., a number of, so CDC, USDA, FDA are putting together a data set that's going to be available about the same time as a ASM-sponsored conference in Washington, D.C. And that would serve as a common data set for bioinformatics developers and other groups to analyze the data, and then they'll be allowed opportunity to show what that data, how their pipelines worked on that common data set, and whether they got the same results as other groups and FDA or CDC.
We have another question in the back. Yes, Stephen Lyon, Sealed Air Corporation. Jack Gilbert at Argonne National Laboratories is helping to coordinate the Earth Microbiome Program, and his lab alone has already processed over 30,000 samples across the planet. And one of the things he's advocating for is the say, standardization of methods. And so all the way from, say, collecting a sample, doing the DNA extraction, up to the processing and metagenomic analysis. And I'm wondering, from your panelist perspective, what would it take to develop a standard set of protocols? Because if you look at certain strains of E. coli and just look at them real mean, they'll lyse. Whereas if you look at something like Mycobacterium, it takes very, very rigorous methods. So I'm wondering, from your panelist perspective, what would be needed to develop a common platform for extraction and analysis?
Well, I mean, I think I just want to draw upon what we're starting to do with ISO. ISO, I didn't get a lot of opportunity to really talk about ISO during the presentation, but ISO is an internationally recognized process for determining consensus and in this case, food microbiology. This is Technical Committee 34, Subcommittee 9, and last year decided to develop a standard for whole genome sequencing. The question that you raise about extraction, I think it's going to be a knotty one, because you're obviously not going to find a common procedure for extracting DNA. And to my mind, and it remains to be seen whether this is where the work group will go, is that's kind of out of the scope. And really what we got to look at is quality assurance programs, what the data looks like. So every bug, every organism is going to have particular ways to extract DNA, have different ways of, there might be different chemistries out there. There are different chemistries out there for determining the data, but ultimately is going to turn into GATCs. And what we have to do in terms of standardization is to focus on the GATCs and the quality of that data.
I think I would agree with that completely. We, in our laboratory, we have a sort of standardized protocol for gram-negatives, and we use a automated DNA extraction. We do also for the gram-positives, but there needs to be a lot more manipulation before they go on to automated DNA extraction. And I think it will be very difficult to get a single standardized method, but I would agree. I think it actually, what's important is the quality of the sequencing data that comes out of it.
And so from a non-government answer, this question doesn't keep me awake at night, thankfully. I'm no longer in the government, so I don't have to worry about these as much. But the reason why is that we've done a fair bit of analysis and comparative analysis looking at different sequencing platforms, different data handling, different references on the same sample set, and typically come up with the same answer. So I'm not overly worried that, I mean, clearly we do want to have standards in place, and we want to have benchmarks, and we want to be able to identify which tools are used and make sure that that's understood. But I still think that if we follow use the commonly used tools, techniques, methods that are out there, we're going to essentially come up with the same answers. Now, when you have to solve whether or not one food producer is actually the source of a contamination, or a medical product is the source of a contamination, and that results in death, absolutely those standards are going to be necessary. But just as an example, the investigation I talked about with the fungal meningitis, those samples were looked at CDC as well as at our laboratory, two completely separate laboratories using three different sequencing platforms, using completely different methodologies for SNP discovery and analysis, and essentially came up with almost exactly identical results. In one case, within the outbreak set, we found a total of 12 SNPs, and in another case, we found 8 SNPs, and that difference of 4 SNPs was then verified and looked at using Sanger sequencing, and we were able to show what truth was. And I'm not going to mention which lab had truth, but they were essentially identical and still had exactly the same results, that the patients in the medical product had identical genomes in them.
So we heard in the presentations several examples of how looking at highly related, and one example there was 2 to 3 SNP differences, another one around 20 SNP differences, and then a third example where it was around 300 and not highly related. So my question is there a definition for highly related?
In an epidemiological sense, I would say no. It depends on the organism, the clonality of the organism, and it depends on the epidemiological question you are asking. Two outbreaks, no 2 outbreaks are completely identical. An outbreak that has a point source where you have a product that comes out once that is contaminated, you won't expect much difference. And if you have a clonal organism like Salmonella enteritidis, the difference might be less than 2 SNPs or something like that. Whereas if you have another outbreak where you have a persistent problem in the production environment, a chicken house, for example, that has been going on for 2 years, well, there we have seen up to 40 SNP differences in the related strains. So you really can't define a hardcore SNP, or for that matter, also if you're looking at MLST data, hardcore allele difference criteria. You have it all depends on the conditions.
Do we have any other questions from the audience?
Hi, my name is Sean No, USD ARS. I have a question for Dr. Smith. And in your talk, you talked about this ANI 0.95. If it's, the index is over 0.95, you consider that one species. If it's lower than that, that would be different species. Is that my understanding? Is that right?
I would say it's probably mostly right. If you look closely at the slide that I showed with the Listeria example, actually between the different lineages of Listeria monocytogenes, you might get somewhat below 0.95. I think we get down to 0.94 and 0.93 or something like that. But it's still the same species for any practical purposes.
Like the known E. coli genome range from 3 point something to 5 something. So are you trying to call those different species, or?
No, well, yes, I'm calling them the same species because where they differ is in the accessory genome. When you are looking at the ANIs, you are only looking at the conserved part of the genome. It's only the regions that are homologous between the 2 strains that you're comparing.
Okay, you are only looking at the homologous.
Yep.
Okay.
Hi, folks. Ed Dudley from Penn State University. First of all, this was a great session. I really hope there will be a Rise of the Genomes 3 in St. Louis. But anyway, so we've heard about a number of different sequencing platforms. Seems like Illumina is pretty popular. Some folks are using PacBio. But I guess I was thinking about into the future. Are any of you folks using any of these, if you will, the next generation sequencers, particularly thinking the nanopore technologies, and what are your experiences with them, and do you think they'll eventually supersede what we're using today?
I said in my talk that we had got the MinION, Illumina MinION, and we have used that in the Salmonella outbreak, the first one I described, the phage type 14b. That local outbreak was in Birmingham. They had a MinION at the University of Birmingham, and they did all their sequencing locally of that hospital outbreak, and they could see they were all identical, but they couldn't, because they didn't have a big enough database, they couldn't put them in the national context. So they sent the sequencing files to the reference lab, and we were able to put that in context. And one of the things we're looking at the MinION is doing is still, even though we've got very good Illumina machines, it still takes quite a while to do the sequencing. So, it's a couple of days, but you've got to do the extraction, whatever. So we're looking at using the MinION, once we know we've got an outbreak, for isolates coming in, to sequence them very, very quickly. So I think definitely, changes in technology will come along and will be adapted, just because it's such a fast-moving field.
So, and then on the fourth generation machines, the nanopore and whatever else is out there, we do not have those in our lab, but we do have collaborators working with them. Primarily the concern is the really high level of error rate right now, but we're absolutely certain that's going to come down. It's a major focus. We already know that it's come down. The ion torrent used to have that problem, and that's starting to resolve itself. And so I think that these issues, we really don't know where it's going to be 2 years from now. We can't even imagine essentially where it's going to be 2 years from now. We couldn't imagine where we are today 2 years ago. So it's just one of those, we got to hold on and try to stay out on front of the wave and keep testing them. But just know that, yeah, some of the newer technologies, they're not ready for prime time, but that's okay, they will be.
We're working with the MinION in College Park, and we're part of the MAP, is the MinION Access Program, and it's been very difficult to keep up with the amount of change, the technology development at Oxford. But we think that the MinION and possibly the Roche instrument that's coming out could be very useful, specifically, as you mentioned, for rapid sequencing. Even with a poor, an error rate, if you have a long read, that could provide, very high confidence of particular target. So we're very excited about these new technologies as they come out, and we look for opportunity to evaluate them.
I think because of all these developments and the technology that are happening, therefore also in the ISO working group, we decided not to take this into the standardization and leave this open, isn't it, Peter? That we know there will be changes of technologies and so on, and therefore it will be different sensitivities, so probably maybe DNA extraction protocols will be different as well. And therefore, also I agree that the proficiency testing would be a better way to compare and benchmark everything than setting really a defined protocol.
Yeah, sure.
Just a quick question about, for organisms like Listeria and Campylobacter, many labs are likely going to continue to use tools like MLST for routine applications. Are the genome sequence data going to inform the MLST databases? I know one can go manually and do it one by one, but it would be nice with 1,500 Listeria strains if one had a way of knowing, say, with the metadata that are there, what MLST sequence type an isolate is, because that can be extracted off of the whole genome sequence. And another related question, well, actually not too related, is if two isolates were to have very highly conserved backbone, but let's say one lost a plasmid, you had how would they figure in the scheme of relatedness?
I can ask first with the MLST question. I think every laboratory that is doing MLST and who owns a sequencer is today doing MLST using whole genome sequencing. It's cost efficient. It's cheaper to sequence the whole genome than it is to do 7 PCRs and sequence all those sequences. And the other question a plasmid. Well, that depends on the analysis that you're doing, because the plasmid is part of the accessory genome, and often when we are doing the comparisons, we are eliminating or we're filtering out all the accessory genome.
But I was going to ask, when you do the whole genome sequencing, you would, of course, also find the plasmid sequences, right? But in some strains, they would be lacking. But they would still be considered, if the SNP numbers in the backbone were below a threshold, that absence or presence of the plasmid wouldn't figure in differentiating the strains. Am I correct?
Yeah, but there's certainly a number of bioinformatic tools that allow us to identify what's present and absent within these, and certainly if we know that they're plasmid or genes that are typically found on plasmids, we can identify those. You can actually just pull out the samples that you want to look at that all have those and compare those, consider all that part of your core genome now, and just compare it among them, look for SNPs inside those plasmid genes as well. So there's a number of different ways you can approach that.
I think we have another question.
Yeah, maybe a totally different question. I'm part of the advisory board of a European project, Decathlon, yeah, and it also looks at whole genome sequencing, but not only for pathogens, but it's a project that looks also at GMO and in customs. And one of my colleagues in the advisory board is actually from US Customs, yeah, and they were very interested to use sequencing also to have endangered species, plant species, if they come in. So the discussions there were like also on validation and more like minimal performance characteristics. So I had the impression they had it quite figured out already in GMO, how they approach this, and that the question is, we're now here food safety and pathogens, but you guys have experience about looking over to other applications which are not food safety, but which are also sequencing, and can we learn from that? Maybe we don't have to reinvent the wheel.
Yeah, we have a very a big project at FDA CIFSAN on using sequencing of fish barcodes, so identifying fish species. And this is important in the regulatory world, because fish goes through a lot of it goes through a lot of pathways of, from the ocean to the market, and a lot of fish is subject to economic adulteration, where you have a species that's worth a lot, and they substitute with a species that's not worth as much. So identification is very important to determine those instances of economic adulteration. Another area which is a little more controversial that's been in the news recently is with the supplements and using barcodes for DNA sequences for supplements, and that is a little bit more problematic and probably needs to be subject to more stringent analysis and quality control.
But the question is, I mean, we're talking about quality control and things. Can we learn from how they do it in these disciplines? Maybe that we don't have to start all over again.
Yeah, I think you've got a very good point, and I think sometimes we are not very good about learning from other areas, other people who are using the same techniques that we are, and if they have already established things like you're saying for quality control, we do need to learn from other people, because I think particularly because it's so fast moving that you're wasting time by reinventing the wheel. So I'm not quite sure how exactly how we do it, but I think you make an excellent point there.
I think so too, and 90% of what we work on is not food protection, and so the way that we've advanced our science is just learning on genomics, primarily from the human genome side. They were way ahead in genomics than on the public health side, but we've also learned quite a bit in forensics in how to handle samples appropriately and for proper analysis. So I think, yeah, agree, you're right on.
I think we have time for one more question, if anyone has. Okay.
Hi, I'm Kanika, working at Cornell University. in the Division of Milk Quality Improvement Program. I did my PhD in dairy biofilms and a bit of whole genome sequencing on Geobacillus, which is a thermophile quite prevalent in milk powders. My question to either of the speakers is that I have seen that whole genome sequencing is more beneficial, it's a culture-based diagnostic methods, where you pick up a single colony and extract the total DNA. Now, do you think it is actually a challenge to study dairy biofilms, especially, not really dairy biofilms, but biofilms formed in the food processing industry? Because that's what the curiosity lies, is you want to see what's the proportion of the isolates formed in biofilms. And technically, we do traditional microbiologies to grow biofilms and then do a plate count and isolate colonies. But do you think it'll be interesting to do metagenomics or whole genome sequencing straight on films scrapped from the equipment and then send it off to the sequencing platforms? And considering that these films also have a lot of lipids and other polysaccharides which might interfere in the PCR chemistry. And it's a long question.
I'll just say, yeah, absolutely. I think it's interesting. I think that it's a great question. And I think that here's where the power of whole genome sequencing will eclipse standard microbiology. You can look at the entirety of the organisms present, not just selectively identify what's growing best on your plate, but see what's growing within the whole biofilm through metagenomic processes. Certainly there are things to overcome, such as inhibitors. But I think that this is where we're going to find the answers as to what's actually growing there. How does that change over time? What's the best way to treat or mitigate situations? Because we can get a complete kind of holistic genomic look in samples, just as what you're talking about.
Thank you.
And well, you're talking about metagenomics here, and I will just then say that there is a session tomorrow. I think it's called I Want It All, I Want It Now, tomorrow at 10:30 or something like that. You should go to that one.
All right, well, I just would like to thank our speakers one more time. It was excellent presentations. And thank you all for attending. This has been a great symposium.